Hi everyone, this brief video is just to remind you of the important parts regarding lipids, very important biological molecule. As always, just thanks to OpenStax for letting us use their slides and for making textbooks free for all. The important thing I want to remind you of for lipids is that all of them are in some way hydrophobic, that is, they do not interact well with water. One of the most well-known examples of a lipid is a fat molecule, also known as a triglyceride. And this molecule is basically made up of a glycerol unit, fused in this case to 3 fatty acids. So remind yourself, before we were talking about bonds that bring molecules together in the context of monosaccharides becoming polysaccharides, that bond is also at work making fat molecules. So in this case, the dehydration reaction would fuse together a glycerol and 3 fatty acids. The specific name for that bond is an ester bond. So you'd see an ester bond here, an ester bond here, and an ester bond here. So when I say that, I'm referring to the dehydration reactions. So this would produce 3 water molecules because we'd be adding this fatty acid, and this fatty acid. So a glycerol fused to 3 fatty acids makes up a triglyceride, and the biological importance of that is, well, you can store a lot of energy in a molecule of fat. In addition, for many organisms, accumulation of fat is one way of storing heat inside the body. So stearic acid here will illustrate an important principle, and that is that this is a saturated fatty acid. So all of the carbons here are surrounded by hydrogen. They're all single bonds. So in this case, the carbons are saturated with hydrogen, so we call it a saturated fatty acid. Whereas with oleic acid, you see here that it's unsaturated, so it's not saturated with hydrogens because of this double bond occurring here between these 2 carbons. There's a lot of discussion about saturated versus unsaturated fats. When it comes to nutrition, I don't necessarily care about that for this presentation. But I will show you what is interesting is that when you're looking at a saturated fatty acid, it sort of presents itself in a linear fashion, whereas an unsaturated fatty acid, because of the double bond, would kink that fatty acid tail there. So when the hydrogens are on the same side of that double bond, we call it a cis unsaturated fatty acid, whereas when the hydrogens are on the opposite side of the double bond, we call that a trans fatty unsaturated fat. So a lot of discussion in the news lately about trans fats. So when we're talking about trans fats, in many cases that is a unsaturated fat that's been saturated with hydrogens through a chemical process, which I won't go into here. Another example of a lipid in nature are waxes. And so interestingly enough, for many leaves, you would see a waxy coating on the surface. So this waxy production of the leaf, it's actually an adaptation in nature because the waxy coating prevents the leaves from losing too much water. So there's an important biological application for waxes, which are lipids in nature. Lipids can also be steroids. So in this case, steroids have a very obvious structure. They usually see these 4 fused rings, and from there you can see various functional groups added. But essentially, you'll see a 4 fused ring structure for steroids, like cholesterol, like cortisol. And many important hormones are also derived from these steroids. So estrogen and testosterone are examples of hormone steroids derived from cholesterol. But for us, the most important lipid that I want to talk about is a phospholipid. Okay, so phospholipids are interesting in that they have a dual nature about them. So portions of the phospholipid are hydrophilic in nature, whereas other portions are hydrophobic, helping it to fit into the category of lipid. So these structures shown right here, and in this cartoon model are the fatty acid tails. So you can see here, this is a saturated linear fatty acid tail, and this is an unsaturated one that has a kink in it. So this polar head region is water loving. That is due to the phosphate that's found on the glycerol, meaning it will interact well with water, but the hydrophobic tails, the fatty acid tails, retain their hydrophobic nature and do not interact well with water. That's important for us as biology students because when you put in a bunch of phospholipids together, they will behave in a manner that's reflective of their biochemistry. So in this example here, you'll see the polar head groups orient themselves out here towards water. Likewise, on this side, you'll see them orient towards water, but the inner hydrophobic core will orient themselves towards each other to avoid water. And what that does is it gives us the formation of a phospholipid bilayer, a cell membrane. So that's really important for living things, and really important as we turn our attention here shortly to cell biology. So just to recap, lipids are a diverse category of organic molecules. They all contain carbon, and they're all very diverse in structure and function. So they don't fit into like a certain category of structure. They're all very diverse. They do lots of different things. And the last important note is that they're technically not a macromolecule, so they're not monomers in nature that you can build upon unit by unit to make a polymer, as you can for carbohydrates, proteins, and nucleic acids. So it's not technically a macromolecule, but it is definitely a very important organic molecule. And these phospholipids that I just mentioned will be, of all the examples mentioned, the most important example for us moving forward. Thank you.